This is NNI Bites Big Learnings, a podcast where fellow healthcare professionals converse about the latest on nutrition science. Join us as Flavia Indrio and Christopher Stewart share their knowledge and insights on microbiome development in infants.
Hello and welcome to today's special episode of the Nestlé Nutrition Institute podcast, NNI Bites Big Learnings. I'm your host today. I'm Flavia Indrio, and I am Professor of Pediatric at University of Salento, Lecce. I am doing research on probiotic in gastroenterology and nutrition in children, and also focus my research on first 1,000 days and non-communicable disease. Today we will be discussing microbiome development in preterm infants. Microbiota colonization even in infants is a very complex process, and specifically in preterm neonate is more critical due to the critical condition of its children since their birth. Today we have a special guest that is Professor Christopher Stewart, and that is the Chair of Human Microbiome Research at Newcastle University in Great Britain. Hey, Chris, please introduce yourself and to our listener and exactly tell us what inspired you to specialize in this field.
Well, thank you very much for having me, and thank you very much for that introduction. So my background is as a microbiologist. I started my PhD looking at preterm infant microbiome just over 15 years ago now. And at that time, microbiome really wasn't the sort of buzzword it is now, I suppose. The techniques we were using were very different. I spent a lot of time in the laboratory, but the techniques have moved on so much now that my lab spend most of the time on a computer analyzing the data. And ultimately, that's why I think the microbiome has captured the interest of so many across many different fields of science, because the data generation now, the level of our understanding is unprecedented. And the challenges for us as researchers is how do we use all of this information? And ultimately, how do you improve the outcomes for those that are the most vulnerable in our population, the preterm infants?
Okay, Chris, can you explain if the preterm infant microbiome differ from the one of their newborn? And if it's so, why is this?
That's a really good question to get us started. And the short answer is yes, the microbes that colonize preterm infants are different to that of normal, healthy term individuals. And maybe we can unpick that a little bit further. But just before we get into that, if anybody hasn't heard of the microbiome before today, this essentially is referring to the collection of microbes, bacteria, archaea, fungi, viruses, and others that colonize a given environment. So my research really focuses on the gut or the intestinal microbiome. And the reason for that really is because that's the largest density of microbes anywhere on the human body is in our intestines. And it's also where most of the immune cells are located as well. So there's this really critical epicenter of microbe immune crosstalk happening all the time. And especially from birth and over those initial days and weeks and months of life, when the bacteria are training the immune system, that's providing not only short-term, but much longer-term health. And so that's really why we're interested specifically on the gut. And when it comes to the comparator with term infants, the microbes differ primarily because the environment and the circumstances of the clinical care of a preterm infant. So preterm infants and those born less than 32 weeks of gestation, they are therefore housed in intensive care units, in incubators, where they have really limited environmental exposure to microbes that term babies would have. They don't go home immediately and get introduced to the cats and the dogs in the household and all the microbes coming from all these other environments outside of the hospital. But rather, preterm infants have much higher colonization by hospital-acquired bacteria. And this is amplified in preterm infants as well, because they're more likely to have antibiotics, more likely to be delivered by cesarean section, less likely to receive adequate volumes of maternal breast milk, and many, many other factors we know impact the microbes and the development of the microbiome. And so this is really important to understand. And as I said, a really good question actually to kick us off today.
Yeah, thank you, Chris. And so after that, we understand that this colonization is so different between the term infants and preterm infants. Is this colonization contribute to health or disease in these two kinds of population, especially focusing on the preterm population?
So we think it does. A lot of what I suppose the state of the field at the moment is association. And so it's really important just to caveat that, as I mentioned earlier in the introduction, we have all of this data, we have a lot of associations, but this isn't necessarily causality. So just when I'm talking about some of our findings, just bear that in mind, that there's still a little bit of a way to go to finally determine exactly how these bacteria may be contributing to disease. But ultimately, there does seem to be some really nice evidence that the microbes actually play important roles in health, less so disease. So again, I mentioned 15 years ago, I started my PhD in this area, trying to really set off on a journey of what microbes are causing a disease in preterm infants called necrotizing enterocolitis, the leading cause of death in this population and a really important disease to better understand. And naively back then, I thought I have 4 years now to do a PhD and find the bacteria causing this disease and win my Nobel Prize and move on. But sadly, it became very apparent very quickly that this is far more complex and nuanced than just a single bacteria causing a disease. But I think to my surprise, and really where the research has taken me since then, is there does seem to be very clear signatures of health. And so babies who typically don't develop diseases like necrotizing enterocolitis have a microbiome that has a lot of bifidobacterium, a lot of bacteria like Clostridium, and less of the sort of hospital-acquired bacteria that I mentioned earlier. And so again, I can't say whether they are causing the health in the infants or simply they are just reflective of a more healthy infant in general, because the research is still, as I said, at the association level, but there does seem to be very clear associations with those microbes and health.
Yes, thank you, Chris. And you mentioned before the role of this breast milk and how these preterm babies sometimes are struggling to get breast milk. But are there bacteria in breast milk? So what about other components of the breast milk that can modulate the infant microbiome?
Actually, this allows me just to mention as well that breast milk is the single most important protective factor against diseases like necrotizing enterocolitis. So rightly, there's been a lot of research focused on understanding what is in breast milk and how that might be leveraged to improve the health of babies who are unable to receive, as you mentioned, the sort of adequate volumes they need. And so to answer your question, there are definitely bacteria in breast milk. This is now well known in the field. These bacteria are there and they're viable. The best way to test that in the lab is if you culture breast milk, can you grow bacteria? And we can. And so again, we know not only are they there, but they're alive. And where those microbes come from, the origin of the microbe is sort of still being studied. We know in the case of direct breastfeeding that the infant's oral community will seed the milk during that feeding process through backflow of milk back into the breast. In the case of a preterm infant, they are receiving expressed breast milk. So that for the most part, so that isn't necessarily happening, but there's still microbes on the maternal skin, on the breast, for instance. And so these microbes are likely also being transmitted into the milk during expression. And I would say just in the context of milk microbes, we have done a lot of work on this recently. Actually, this is all unpublished data, but we don't find any strong link between what microbes are in milk and the outcomes of those infants. So the microbes themselves don't seem to be contributing in a very obvious way to health or disease, but they're definitely there. You mentioned the other components. This is, I think, where the story gets really interesting. And so I think what we've got a much better handle of over the last 2 or 3 decades is what is in maternal breast milk. And a lot of the components that are really abundant actually exist to feed the microbes and not necessarily there to feed the infant. And an example of this is human milk oligosaccharides. These are really abundant sugars in breast milk that have no nutritional benefit to the infant at all. So the mother produces these sugars into their milk at huge energy costs. So what are they doing? Ultimately, they're feeding what we think are good bacteria, things like bifidobacterium and others, using those sugars to colonize the infant gut and in turn produce a lot of really important products that contribute towards health. That's the best of our understanding today.
Thank you, Chris. So it's so important, this kind of colonization. But can probiotics be used to improve this health in preterm infants? And we heard a lot during the last days, last weeks about the risk of sepsis. So there are other options to remove this risk of probiotic sepsis in preterm infants.
Yeah, probiotics, I think, are one potential therapy for use in preterm infants. So I mentioned earlier, they have this abnormal microbial colonization that we typically wouldn't expect in, for instance, a healthy term baby. And so the obvious question would be, why don't you just take those microbes that we do expect in healthy term babies and sort of artificially provide them to preterm infants? And that's exactly what the probiotic therapy is doing. So I think when we started to publish on this, again, maybe 10, 15 years ago, there was increasing interest in using live bacterial therapy or probiotics in preterm infants and providing these sort of so-called missing microbes. And our unit here in Newcastle introduced probiotics over 10, maybe now 12 years ago, and have continued to use them ever since. And many other NICUs around the world in both industrialized and non-industrialized nations have begun to use these as therapy. So I think overall, meta-analyses support their use in preterm infants, is my opinion. The pros outweigh the cons, the good outweigh the sort of risks of probiotic therapy. But as you mentioned, there are certainly risks involved in this type of therapy. And the obvious one is, what if in these immature infants with immature gastrointestinal tracts, some of these probiotics can translocate through from inside the gut, where bacteria usually live, to on the other side, the submucosa and the more sterile sites of the human body where microbes shouldn't be. And this is definitely a risk of providing a probiotic that can happen. This happened recently last year in the US, and very sadly, we had a case of fatality due to that probiotic sepsis. And on the back of that, the FDA, the Food Drug Administration in America, unconditionally said that they should not be used in neonatal intensive care. So this has really spawned a huge debate in the field. And certainly in the US, colleagues are moving away from using probiotics altogether. The biggest unknown for me that we'll never be able to answer is, ultimately, that baby was clearly immature and premature and may well have got sepsis regardless of whether they had probiotics or not, but the bacteria that caused that sepsis may have been different, and then obviously wouldn't have been a probiotic bacteria in that case. So as I mentioned, I think the main benefits still outweigh the risks of this. The meta-analyses support that they can reduce diseases like necrotizing enterocolitis, and much more work ultimately needs to be done in this area to understand what's the best probiotic to give a baby, what's the right time to give it, how much of the probiotic should we give, should we change which probiotic we give depending on the status of that individual, maybe reflecting their diet or their birth mode or many other factors, their gestational age. So ultimately getting towards precision medicine. What's the best therapeutic probiotic for the baby at the right time ultimately?
Thank you, Chris. So can you tell us and just explain us a little bit in detail which kind of research are you doing in your laboratory to better understand how the diet and the microbiome impact on infant health?
Yeah, so we do a lot here in Newcastle unpicking this concept entirely. So we do a lot of microbiology. As I mentioned, my background is as a microbiologist. It's where I feel most comfortable. And this is really kind of basic science. And I mean that in the sense of fundamental science, where we culture bacteria from infant poo samples that we collect here as part of the Great North Neonatal Biobank. I'm lucky to work with fantastic clinical colleagues who really, through their passion and dedication to improving health in preterm infants, have generated a huge biobank. So we have over 100,000 samples now collected from preterm infants over the last 15 or so years. And what my research group can do is take those samples, try and isolate the bacteria from those samples, and then see what components that are in breast milk, for instance, HMOs, or human milk oligosaccharides, as I mentioned earlier, but many other components of breast milk and what we call sort of prebiotic components are in breast milk. How are they supporting the different bacteria? And that just helps us better understand if certain bacteria are really abundant in infants. Is it because they are receiving breast milk that have particular prebiotics that support their growth, and allows us to get much closer to the sort of detailed, nuanced understanding that we need for understanding microbiome trajectories. And the other, I suppose, newer thing that we're doing in the lab is using a technology called organoids. Now, I started to learn organoids when I was a postdoc in the US several years ago in the lab of Mary Estes at Baylor College of Medicine. And I do believe this is going to be a really important technology for better understanding how microbes from humans change human epithelial function ultimately. And the type of work, what I'm getting at, is that you can't do easily in animal experiments. It's really hard to add human microbes to different species that are non-human ultimately. So what we do here in Newcastle, when an infant sadly has surgery and a section of their tissue removed, very often for necrotizing enterocolitis, but maybe milk curd obstruction or atresia or something like this, we can collect the tissue and effectively regrow that baby's intestine in the lab. And we have these stem cells that generate into the gut, the epithelium. We can differentiate. So we have all of the major cell types, and they are functionally active. So we can have different cells like goblet cells produce a mucus. And as I say, I think this is going to be a really important model to add. For instance, what we're doing every day now is adding different bacteria, different stimuli, different metabolites, different breast milk components together or separately to these models, to these organoids, that understand how different patients would or different babies would respond to these different factors. Not every baby responds the same way to the same thing. And so it is allowing us to much better understand this complexity, this sort of individualized response to different components. And my hope, through a lot more work, I must say, we're years away from this, but ultimately I hope this model can provide us a way of better engineering probiotics or postbiotics or ultimately therapy in these infants, doing the types of experiments that are critical to get towards precision medicine, I think.
Thank you, Chris, for let us know all this news and especially to give us some, I can say, hope of the new approach on the health and disease, changing the colonization and the microbe of these preterm infants, because our ultimate goal, as you say, is to care about the health and disease on this fragile baby. Thank you again.
Thank you to listeners for tuning in.
And that concludes today's episode. Thank you for joining us with Flavia Indrio and Christopher Stewart as we talked about microbiome development in infants. We hope you enjoyed and learned from this quick discussion, fellow experts. Till the next episode.